Hi there. It is Jacob. This is Jacob Gibson. Today is May 19th, 2024. This is my video recording or my video presentation for the reproductive system portfolio assignment. For this one, I chose to study how sperm matures in the germinal epithelium. Reason why I chose this one is just it's not something that's usually emphasized in a physiology course. There's not as many clinical applications as there is with the female reproductive system. So it doesn't get talked a whole lot about, but it is a really cool process. Again, just to kind of know about and just how sperm is produced. And this process that we're going to talk about here is called spermatogenesis. Again, genesis means to make. So we are making sperm here in this process. So where this occurs in the semiferous tubules in the testicle. All right, so again, we're looking at the gonad here, which being the testicle. When you look at a testicle, you're going to see these really just kind of winding tubes, and that is going to be the seminiferous tubules. That's where the sperm production is going to take place. As sperm are produced, they are going to move up here into the epididymis. All right, the epididymis, that is where the sperm final maturation takes place. But really today we're just going to focus on what occurs right here in the semiferous tubules. And then you'll get the vas deferens here, and that's going to, again, are going to move the sperm out of the testicle, but I'm not going to focus on that process here. I just want to focus on where sperm is produced. So the seminiferous tubules, the lumen that you will kind of see right here. Again, lumen means just that hollow tube. Right there, the lumen is going to be made out of some smooth muscle. There is going to be some peristalsis that's going to help the sperm move. And then again, like I said, it matures right here in the epididymis. And there's going to be kind of two different things that they kind of get mixed up when you look at this process. And I kind of want to make sure I define them. So I told you this whole process is called spermatogenesis. This is going to include the whole process. And you're going to be dealing with the cell division part. And then there's spermiogenesis. Spermiogenesis is going to be focusing on taking the spermatid and making it into the final part. Spermiogenesis is the final part of spermatogenesis. So sometimes those words get mixed up when you kind of do research on this topic, but I just want to make sure I clearly define that. All right. So I'm going to focus, like I said, on spermatogenesis. All right, just the whole process right here. So again, this is a cutout section of a seminiferous tubule. One thing I want to be just very clear on is this isn't what it actually looks like. It's very, very much more compact, but this just kind of does just the general kind of sense of things. So in a seminiferous tubule, what you are going to see is the main part of this whole process is the main cell that really is going to have a lot of influence is the Sertoli cells. They are right here in gray. And then in between these Sertoli cells, in these tight junctions, that is where you are going to see spermatogenesis occurring. All right. And as the cell, or as these spermatogoniums, or whatever stage the sperm cell is in, as it moves down in that junction, it will mature over time. And in the spermatid stage, you're not going to have any motility in chemical composition. That is going to be the spermiogenesis part of it, not the spermatogenesis. That final stage is going to give it that chemical and motility composition. So what happens here is that you're going to get both mitosis and meiosis occurring. Right? Those are the two types of cell division that's pretty common in all of biology. So in the outer part here of the seminiferous tubule here, in those tight junctions at the top, you're going to see spermatogoniums. Spermatogoniums are the stem cell. Now that stem cell is going to undergo through mitosis. And now mitosis is the process that it's just a copying of the cell. So spermatogonium undergoes mitosis, it makes a copy of itself. Well, you're going to end up with two cells in that process. You do that. One of those cells is going to continue the process in developing to a full sperm, whereas the other cell that is produced is going to go back and become a stem cell that'll be copied again. Okay? Then you generate a primary spermatocyte from that first cell division. You start with the stem cell, the spermatogonium, you go to a primary spermatocyte. You are now going to undergo meiosis. Now, in meiosis, it's similar to mitosis, except for you are going to be going from a diploid cell to a haploid cell, so that when it meets the female haploid cell, the chromosomes line up. Okay? So in meiosis 1, or I should say this primary spermatocyte, it's going to undergo meiosis, go through meiosis 1. Now, meiosis 1 is basically mitosis, except for you're going to have some crossing over events of the chromosomes, and the genetic information is going to get a little bit mixed up. That's why you don't look exactly like your siblings, is because information gets switched up and exchanged. And it's, again, a really good advantageous with any type of sexual reproduction. You want to have genetic diversity. So that's what's going to go on there. In meiosis 1, you get that crossing over event. It's another copying, but you're going to end up, and actually I don't like this one because you technically end up with another diploid cell at the end. You're going to then go through meiosis 2, which is going to make that diploid cell haploid, and it's going to make sure the chromosomes only have one copy of each other, so that it can meet, and you're not the other female cell, and you're not going to have any crazy duplication events. And then the last part here, which I'll go into in a little bit, is going to be the spermiogenesis. That's where you're going to have the cell differentiation. You really just have a cell here. Later on, you're going to give it kind of those sperm characteristics with the tail and the head and all of the parts you kind of associate with a sperm cell. But big picture here, again, you've got these stem cells. They move down and they go into the lumen. So the cells that are closest to the lumen are going to be the ones that are most developed. There's also these Leydig cells up there. Really, they're just there to secrete testosterone. I'll talk about those in a little bit. So this is the kind of the hormonal control of the process. You don't spermiogenesis pretty much occurs throughout life, but really where you see it to start take off is during puberty. In puberty, you're going to have gonadotropin releasing hormone. That's going to be secreted from the hypothalamus. That's going to travel down to the pituitary gland. That's going to stimulate LH and FSH, similar hormones that you see in the female reproductive system. LH is going to mainly kind of work on the Leydig cells. That's going to secrete testosterone, which is going to influence sperm production. FSH is mainly going to go to the Sertoli cells. Again, those are those cells that line the whole thing. Again, this isn't accurate. They're really, really compact in there. This is where the cells that are going to nourish those spermatids as they go through development there. So, and then again, as there's a negative feedback loop here too, because as these things develop and as these hormones are secreted, eventually inhibin was going to be released. That's going to stop the production there. So the more production you have here in the Sertoli cells, then you're going to have inhibin kind of stopping the process. So those are kind of the main hormones. It's very similar to the female reproductive system. Like I said, the Leydig cells, that's going to produce your testosterone. That's going to be influenced by luteinizing hormone. There's again a few other things that can impact this process. The environment can play a big role. Sperm does not like warm environments. So again, if you're dealing with a lot of warm or maybe cold environments, you're going to see a decrease in sperm production, as well as dietary impacts. Worldwide, we are seeing a decline in male sperm levels. Again, that could be a problem because of obesity and things like that in our diets. that we're not producing enough of these hormones, and there's some other issues going on. So, and that's something that's being a little bit more studied. And then again, over time, we do see a drop in, or I should say, this whole process occurs throughout life, but we do see a drop in older individuals. They do become more infertile over time, but they still produce sperm. So again, like I said, going back there, you have those meiotic, mitotic divisions, and that's in the spermatogenesis piece. But the final stage is we're going to take those copying of those sperm cells that are now went from diploid to haploid, and we are going to make it into a full-on sperm. So this is the final stage, is the spermiogenesis. Again, the sperm cell starts very spherical in shape. It has a nucleus, Golgi apparatus. It looks like kind of like a cell that you would consume, but then we're going to make it so it is more motile and can do its job of fertilizing the egg. All right. So again, we're now in the later part of those Sertoli cells moving down in those junctions. So there's different 4 phases. The Golgi phase. So all these phases kind of are named after a certain organelle, because what I think that's really cool is because all the organelles are going to be kind of reused in that spermatid that the organelles are going to be rearranged. So first, the Golgi body. Again, when we first start out the cell, it's pretty pretty same throughout, but it is going to become more polar as we move through. The Golgi body is going to start to make the acrosome that we'll see in the sperm cell. That's going to be right near the head. So when that sperm cell penetrates the egg cell during fertilization, that acrosome is basically just going to release a bunch of digestive nutrients to make a hole so that it can get inside the egg cell. So that's where you start to see kind of the specialization here of the sperm cell. So the Golgi phase is first. You start seeing the development of the acrosome. The Golgi body, do that, you're going to start seeing the mitochondria gathering in different spots. And then you go through the acrosomal phase. That's where you're going to see the cap. So the cap of the sperm cell, that's going to be the acrosome. That's the acrosomal phase there. Then we go to the tail phase, and this I think is really cool. During this whole process, the centrioles are going to line up near the end, and the centrioles are going to be repurposed. Instead of being an anchor for cell division, the centrioles are going to line up, and they are going to start to form a tail. Really cool. The organelles are being used for different means. All right. So you go through the tail phase, and then you go through the kind of the maturation phase. So you've produced this tail here. And then, again, like I said, those Sertoli cells in the seminiferous tubules are so important. They're going to kind of phagocytose all of this kind of extracellular membrane cytoplasm that would slow the sperm cell down. It's basically kind of a cleanup phase. It eats away the cytoplasm, and then you're basically left with a full-on sperm cell. So again, this process is really cool. It's really cool to start. I was kind of doing some reading at other websites that they were talking about how this process is kind of evolutionarily conserved, that you see it kind of throughout different organisms. It's a process that, like I said, there's a lot of different environmental factors that can influence it. It's something that again doesn't have a whole lot of clinical significance, but it is something that is important to know about, and just how we can focus on development. And later on, we can start to focus on kind of some male fertility issues too, as we understand this process. So that was just a really quick introduction. It was a really fun project. Again, those are my references. Just some photo credits for the photos I found. That is it for me. So thank you.